Hello everyone. In this video we are going to continue learning about chemistry. Specifically, we are going to cover the last of these learning objectives that I gave you in the last video. These ones are all relevant to your study of biochemistry, so the molecules that are specifically important in building up the human body. Again, in the last lesson, we talked about essentially inorganic chemistry. In this lesson, we are going to focus more on organic chemistry. And so really, organic chemistry is, of course, the study of the molecules making up living things. Organic molecules in general are just defined by the fact that they contain both carbon and hydrogen. And we'll see that most of them also contain oxygen as well. The specific type of organic molecules that we are going to be learning about in this lesson are called biomolecules. And so bio is emphasizing the molecules that make up living things. And so what all of these biomolecules have in common is that they have what is called a carbon backbone with functional groups attached. In order to understand this, let me relate these biomolecules with their backbones and functional groups to vertebrate organisms. Okay, so what we can see on this slide is a whale down here, we can see a bird, and of course we can see a human over here. And what they all have in common, among other things, is that they all have a spine. Right? The spine happens to be made up of subunits called vertebrae, which all generally have this general structure. This one in particular is a human vertebra, but in general a vertebra is a vertebra. Okay? So the spine is made up of subunits called vertebrae. Now attached to that spine are of course appendages, right? So a whale has fins, right? Birds have wings, humans have arms and legs. And so what makes each of these organisms function different in the environment, right? So a whale in the ocean, a bird can fly in the air, humans can manipulate things with their hands and run with their legs. So what gives rise to the function of all of these different organisms isn't their spine, right? That's what's in common among all of them, but instead it is their appendages. And so in exactly the same way, biomolecules are made up of a carbon backbone, right? So that's pretty much similar or has patterns among all of the biomolecules. Each of those backbones are made up of subunits, right, that come together like this right here, varying numbers of subunits, of course with some modifications, right, depending on whether you're a carbohydrate or a lipid or a protein. But what really makes the molecules behave a particular way within the organism is the functional groups relative to our appendages that are attached to this backbone. And so what exactly is a functional group? As you can see here, a functional group is a specific combination of bonded atoms that always reacts the same way, right? So we can have we have lists of these functional groups, and essentially when a particular functional group or combination of functional groups attaches to the carbon backbone, this can completely change or ultimately determine the molecule's chemical behavior. So here, for example, are two functional groups, a hydroxyl, right? So a hydroxyl is an OH, right? And whenever it attaches to a carbon backbone or another molecule, here shown as R, right? Whenever you see R like this is a variable. This is talking about any other molecule, protein, lipid, carbohydrate, nucleic acid, whatever you're talking about, that can be R. But as soon as the OH is going to attach to it, the molecule's chemical behavior is going to change. In this case, the molecule is going to become more soluble. Okay, another example here is carboxyl, COOH. Right, we're actually going to see that a few times today in this lesson as well. And so an example of this, here is a steroid molecule. We'll look at these again in this lesson, but what I want you to see right now is these carbon rings. Right? If you have taken organic chemistry or even sometimes in general chemistry, you might be familiar with the fact that every time you see an angle like this, that is a carbon. Right? So each one of these angles is indicating a carbon that is bonded to pretty much all the hydrogens it possibly can be. So carbon has 4 openings to bond with other atoms. So here's 1 bond, here's 2, and so hypothetically there's a hydrogen here and here as well. Okay, so that is beyond the scope of what we need to be thinking about right now. What I want to point out is that steroid molecules have overlapping carbon rings, right? So each one of these rings is a bunch of carbons around in a circle. And because of the nature of these molecules, the shape of these molecules, steroids are generally hydrophobic. That is, they do not mix well with water. They are a lipid, which means that they are non-polar. Okay, and so this particular steroid happens to be estrogen. Estrogen, as you know, is a hormone, and so as part of the endocrine system, estrogen is released into the blood. In the blood, which is mostly water, by the way, it is circulated around the body and ultimately delivered to its target tissues. And now the problem with that is that steroids, again, are lipids. They are hydrophobic. And so in order for a hormone to actually be able to circulate in the blood, it has to have at least part of itself being hydrophilic. Okay? And so that is where these hydroxyl groups come in. Right, so the rest of the molecule here can be hydrophobic, but as soon as we add this hydroxyl group, now this molecule is capable of being in water, and therefore it is able to circulate throughout the body and be delivered to its far-off target tissues. Okay. So again, functional groups are essentially like our appendages. They are what determine exactly how the molecule is going to function. The carbon backbone, right, is also a very important component, right? We can't just be a bunch of appendages without a spine, right? We need the spine as well. And so why carbon, right? So carbon is part of every biomolecule, right? Every organic molecule even, and carbon has this unique role because it really is ideal for forming the backbones of these biomolecules. As I just mentioned, carbon has 4 valence electrons, which means that in order to fill the octet rule, it needs another 4 electrons. And generally, it's going to share these electrons, so form covalent bonds with up to 4 other atoms. Okay? Oftentimes, at least one of these other atoms, often 2 or 3, are carbon-carbon bonds, right? And this particular bond is very stable, which is a good thing, right? We don't want our biomolecules to unravel, to degrade. We want those bonds to be maintained because, of course, those are the bonds that are keeping us together. Okay? Also, carbon often forms double bonds, right? And these double bonds, as we talked about in our last lesson, are also really strong. Again, we need to have stable molecules in order to keep ourselves from literally falling apart. Okay. Finally, carbon is very important as the backbone atom, the backbone element, because it can, of course, interact with up to 4 other atoms. And as a result, this allows very diverse molecules to be formed. Right? So again, we can have very different organisms, whales, birds, humans, all with the same general body plan. Okay, so all life forms on Earth that we know of have carbon-based backbones. Ultimately, the specific pattern of those backbones can classify these biomolecules as one of 4 classes. These are the 4 classes of biomolecules based on, again, that carbon backbone structure. Okay, so the carbohydrates, the lipids, the proteins, and the nucleic acids. Okay, in general, we can classify a molecule based on its carbon backbone, and in order to do so, we would need to zoom in to what their individual monomers look like. Okay, so just like our spines have individual vertebrae which come together to form our backbone, biomolecules have individual monomers. Monomer is the name for the subunits, the general subunit of a biomolecule. So for example, in the proteins, amino acids are the monomer, the single subunit of the protein. When they join together with specialized bonds that we'll get into today, this chain of monomers is called a polymer. Right? So these are the larger biomolecules. This would be the entire backbone. Okay. Each biomolecule class or biomolecule category has different names, right, and specific examples of monomers and polymers. So for example, for the carbohydrates, one single subunit, one single vertebra is called a monosaccharide. So mono for one, sac, right, S-A-C-C-H. Here, this is like a single sugar, and we'll talk about this more in a moment. When multiple monosaccharides are bound together, this forms a polysaccharide. Okay, so today we are going to walk through the 4 classes of biomolecules. It is important for you guys to learn the function of these biomolecules. Okay, so what exactly do they do? Why do they need this particular structure? Of course, how does structure match function? And of course, you should also be prepared to identify a molecule as a carbohydrate monomer, a protein polymer, and so on and so forth. Okay, so just as a very brief preview, the carbs, the carbohydrates, are important in energy, support of our body, as well as recognizing different types of cells. The lipids are important in making up the majority of our cell membranes, so literally separating each of these individual subunits from the outside world and from each other. They store energy, of course, right? Fats are lipids. They send signals like steroids and so on and so forth. Proteins do like everything, right? Everything you can think of in the body that's going on involves proteins. And the nucleic acids store both genetic information as well as brief bursts of energy, which is actually used as our cells' energy currency. Let us start our discussion of these classes of biomolecules by looking at the carbohydrates. As I just said, the carbohydrates provide immediate energy and structure to a lot of different things within our bodies. These are sugars, right? So whether you're eating a candy bar or a potato, all of these have carbohydrates, right? So sugars within them. Generally, these molecules have a chemical formula with this particular ratio of carbon to hydrogen to oxygen. Right? So, for example, a single monomer, right? In this case, a monosaccharide, mono 1 saccharide sugar, a single monomer of glucose, for example. So this is an example of a single monomer of carbohydrates. Glucose has 6 carbons, 12 hydrogens, and 6 oxygens. And we can see from down here that glucose exists in a hexagonal ring. So a hexagon, 6 corners. Remember that the corners that don't have anything written are actually carbons. So if you count these up, there are actually 6 carbons and of course 1 oxygen making up the 6th corner here. Glucose is important because it fuels most of our cellular functions. That is, it is really easy for our cells to take glucose into them and complete a lot of fancy chemical reactions, which we'll get into, and ultimately produce ready energy to fuel contraction of the muscle, generation of action potentials, cell replication, all of the things that cells are doing on a daily basis. Okay. Glucose is, again, what we'll be talking about the most, but there are also a lot of other important what are called isomers. So these are molecules that have the same chemical formula, but a different shape. And so one example of this is fructose, right? So high fructose corn syrup. Right, just a different shape. This is a pentamer, so 5 carbons in a ring. Right, again, each of these corners is a carbon. Fructose is another molecule from which we can get energy. Okay. Take-home message here is that these monosaccharides are always these nice little carbon rings. Another important monomer, right, monosaccharide of the carbohydrates that we're going to be talking about is ribose and deoxyribose. So these are 5 carbon sugars, and they are going to be an important part of both our DNA and our RNA. Stay tuned. Okay, so monomers are ready energy, but for the most part, if we need structure or we need some energy to be stored, we need to start binding these monomers together. So starting to construct a spine out of individual vertebrae. And so the way that we do that is we actually dehydrate these molecules. Right? So what we can see here are 2 glucose molecules. Right? These are the reactants. Right? Note that attached to this carbon right here, there is an OH. right? And so using an enzyme, we can actually remove water molecules, right? So an H from here, an H from here, so there's H2, and an oxygen from one of these molecules, leaving behind a single oxygen in between. And so again, an enzyme is going to remove the water, right? So if we look on the other side of this equation, we can see water. Here's water, sorry. That has been removed, again, by an enzyme. We'll talk about that. And now, where we once had 2 glucose monomers, now we have a maltose molecule. Right? So what I want to point out is that we still have these nice carbon rings, but now they are attached with an oxygen in the middle. And so that's going to be really important. Remember, I told you that steroids have overlapping carbon rings. Carbohydrates have carbon rings in a chain, each of them separated by an oxygen. And so that's going to be really important in differentiating between these different types of molecules. Look for the oxygen between. Okay, and so anytime we want to make a polymer, so chains, big clunky molecules out of individual subunits, we need to complete a dehydration, also called a synthesis reaction, right? So we are literally dehydrating, we are pulling water out, and we are also literally synthesizing a larger molecule. Okay, so what we can see here is an existing polymer, right? Each of these little purple circles here represents an individual monomer joined together by some kind of bond. An enzyme can pull water out, right? And these enzymes are specific to whatever polymer they're trying to make and ultimately create a larger polymer. So to go from monomer to polymer, we are always dehydrating or synthesizing a larger molecule. Okay, so sucrose, as well as maltose, which we just saw a couple slides ago, these are all called disaccharides. Remember, mono for 1 sugar, di for 2 sugars. Right, so this happens to be the only biomolecule class, the carbohydrates, which has a special name for when we stick 2 monomers together. And the reason why we have a special name for the disaccharides, where none of the other biomolecules have a special name for 2, is because these happen to be sweeteners. All right, so we have a special name because we love to eat them, because we love to interact with them. So sucrose, as I just showed you, is a glucose and a fructose bound together. All right, again, using an enzyme to dehydrate the 2 molecules. Note again, there is an oxygen separating the 2 carbon rings. Let's see, maltose, right, that we looked at a couple slides ago, is 2 glucose put together. This one is really important in doing fun things like brewing beer. And lactose is a glucose and galactose, which again is an isomer of glucose, and this is the sugar that is found in milk. Okay, so again, these are all sweeteners, sucrose being just normal table sugar. I also have on this slide a couple examples of artificial sweeteners, right? So sweeteners that can be made in the lab. In general, these things work as sweeteners because they have some component of the molecule that binds to our taste buds, the receptors in our taste buds, and essentially tricks those receptors into sending a message to the brain that, hey, this food tastes really sweet. But at the same time, these have been synthesized in the lab. We don't have the right enzymes to break them down, and so for the most part, we get very few, if any, calories, so very few, if any, or any energy or ATP molecules out of these particular molecules, just because we can't digest them. And so it would seem to be a win-win, right? You get the tastiness, but you don't get the calories if you're worried about, metabolism, energy, weight, whatever. But as with anything, there's no free lunch. There are many downsides to this, and I'm not going to get into that now, but I do give you a nice, easy to read article here from some respected scientists, and I say easy to read because it's written for high school students, but very thorough about how these things can literally change your brain and have long-term consequences on your behavior, including appetite, depression, etc. And that, of course, is in addition to the fact that some of these most commonly aspartame, are linked to things like headaches and cancer and whatnot. Okay, so definitely look into that if you are the type to use artificial sweeteners. Okay, next, the polysaccharides. The polysaccharides are carbohydrate polymers with more than 2 monomers bound together. So a few to hundreds of monomers. What we can see up here is a glycogen molecule. This happens to be what the human body uses to store energy, right? So essentially, if you are sitting on that couch eating a candy bar, you are not using all of the energy that's packed into those glucose molecules, and so your body's going to digest the food into glucose monomers, and then in your liver, in your muscles, and your fat cells, our body essentially sticks those extra glucose molecules that you don't need in your blood, you're just sitting on the couch, you don't need them, it's going to stick them to this big branch glycogen molecule. And so each one of these tiny little circles here represents a carbon ring of glucose or fructose or whatever. Okay, and so when you get off the couch and then go for a jog, then our bodies can take these little monomers back off of this storage glycogen molecule and use them to fuel our bodies. Okay, so again, in animals, we use glycogen as an energy storage molecule. All right, so big, clunky polysaccharide, very branched. In plants, they do the same thing, but with starch. Right, so as they're photosynthesizing, if they're not using that energy right away, they can store the extra glucose that they just made via photosynthesis in these long chains, so not branched globby molecules, but chains of carbohydrates called starch. And so that's important because we, of course, eat starch. Right? So the plant might store these extra molecules within the tuber, within the root, the potato, and then, of course, we eat potatoes because they're so tasty. Right? And so we are interacting with this plant storage molecule in our own human bodies. Okay. As I said before, polysaccharides are also important in structure. So plants use a different type of molecule, and I'll talk about this in just a moment. So a different type of glucose chain to create structure. So that nice rigid cell wall that allows plants to stand up without a spine is called cellulose. And again, we eat plants, and therefore we eat cellulose. Okay, and so we can see cellulose here is a nice chain, and again, carbon rings separated by an oxygen. I cannot say it enough. In humans, right, so in any animal, any fungus, we use a polysaccharide called chitin to give us structure. And so we've all seen this, this exoskeleton being shed here is made of chitin. Okay, and so when you are sitting on the couch eating a candy bar, you are taking in sweet molecules, right? So molecules within the candy bar, like Ted here, into your digestive system. The only way to actually take those molecules into your blood and therefore be able to use them in your own body is to first digest them, right? You can't just like start, putting Snickers molecules all over your body. You need to digest it first. And so the process, right, the reaction that is actually going to allow that to happen is called hydrolysis or decomposition. And so this is literally exactly the opposite of the synthesis reaction, right? So the dehydration synthesis reaction, right? Here we take in big long candy bar molecule using enzymes in your digestive system. We add water, right? So an OH to one and an H to another, and therefore break these molecules apart. And so this individual monomer here can be absorbed across your intestinal lining into your blood and then distributed to wherever it needs to go in your body. And so this reaction is called hydrolysis because you are literally adding water to the larger molecule. Also, it's called a decomposition reaction. When you decompose something, right, you're taking something big and making it into smaller little pieces. Okay, so that's exactly what's happening here, polymer into smaller monomer pieces. You can remember which one is which. If you think about putting a pan in the sink and soaking it in water for a while, right? So those big, crusty food molecules that have been cooked onto the pan too much. In order to get them off, you soak them in water for a while. And so even though there aren't enzymes in the pan, given enough time, water will automatically start breaking these bonds and therefore make it much easier to wash away these smaller molecules. Okay, so hydrolysis is going from big to small. Dehydration is going from small to big. Okay, so I do want to talk a little bit more about the different polysaccharides of carbohydrates, because of course we eat both of them and they are handled very differently within our body. Do plant polysaccharides, that is. Amylose is starch, essentially. Okay, so eating a potato, we can obviously get lots and lots of energy, for better or worse, out of potatoes. But when we eat cellulose, right, so essentially the rigid plant walls, so eating celery, eating your fiber, this is when you get glucose. Okay, so amylose, of course, is digestible, lots and lots of energy from starch. Cellulose, for the most part, is not. Right? So it's not digestible by most animals, or any animals. It is, well, because it is very difficult to break down, it's really good for making fibrous things like paper, clothes, and whatnot. Right? But we're still told to eat cellulose, right? And we'll talk about that here in just a second. First of all, why can we get lots of energy out of starch, but we can't get any energy out of cellulose, right? Energy from potatoes, not from fiber. Okay, and the answer to that is actually in this bond angle right here. Right, so here we have two carbon rings, so a portion of this polysaccharide. Note that both of them have an oxygen in between them, but the angle of the oxygen bond is different between the starch on the top and the cellulose on the bottom. With starch, there's about a 90-degree angle. With cellulose, there's about a 180-degree angle. Okay, so that makes all the difference in the world. And it makes a difference because each enzyme, as we're going to see, is capable of breaking only a particular type of bond or angle of bond. And so amylose, right, so starch here, can only be digested efficiently, of course, by amylase, right? So we'll see that the suffix ase means enzyme. And so amylase can recognize this about 90-degree bond angle and therefore break amylose into maltose, right? So these two two monosaccharide molecules called disaccharides. Maltase is the next enzyme, which breaks maltose down into 2-glucose, and of course glucose is what is capable of crossing over our intestinal lining and being used by our body. And so the human body, right, and all mammals can produce amylase. We have the gene that codes for this particular enzyme, and so we release amylase into our saliva and start breaking down starch right away. We release maltase into our intestines and then break down these molecules the rest of the way. And so we can get glucose, which is what we're actually looking for, because we can produce both of the enzymes required to recognize these bonds. Now, cellulose, on the other hand, requires different enzymes, right? So the amylase cannot recognize this 180-degree bond angle, but cellulase can. Okay, and so cellulase would then break the cellulose into disaccharides, and cellobiase breaks those disaccharides into glucose. Okay, so the end product is the same, but the route for getting there is completely different. And so the problem with a human trying to eat fiber and expecting to get any energy out of it is that we don't make this. We do not make cellulase, and as a result, we cannot break down cellulose and actually get glucose out of it. Other things can. Okay, so this is one of the many functions of your gut flora, and I hope that we can talk a little bit more about this as we go along. But no animals can produce cellulase, but most microbes, particularly bacteria and fungi, are capable of producing cellulase. And so animals, some animals, particular herbivores, spend their lives eating grass, eating this super fibrous material that is just chock full of cellulose, and somehow they're getting energy out of it. And so animals like cows have actually developed a chamber of their stomach called a rumen to specifically support this bacteria. And so they actually have a symbiotic relationship. The bacteria breaks down the cellulose for them, and in exchange, the cow protects the bacteria and gives them lots and lots and lots of stuff to eat. Okay, so very beneficial for both. Horses, rabbits, goats, humans, the rest of us have developed what is called a cecum. Right, and so we'll talk, or you'll learn about this a lot more next semester, but the cecum is essentially this outpocketing of our intestines, and essentially this is like a bacteria hotel. Again, a symbiotic relationship. Bacteria produces cellulase, breaks down cellulose. The animal can then utilize the bacteria and or the glucose that it's actually producing in exchange for this nice cushy hotel. Of course, herbivores like horses have an absolutely massive cecum, right? Their lives absolutely depend on this function. Humans are omnivores, and so our cecum is greatly reduced, but we still depend on our gut flora nevertheless. Okay. The next class of biomolecules are the lipids. Okay, and so these differ from the carbohydrates in that the lipids are nonpolar. Right, so carbs are polar, they are hydrophilic, they can easily dissolve in water. Your Kool-Aid dissolves in water. Lipids, on the other hand, do not. Right, so think about the fact that fats and oils separate out automatically from water. Right, so here we can see fats and oils automatically forming a droplet, thus decreasing the surface area exposed to those polar water molecules. On the other hand, proteins and sugars can, of course, easily go into water because they have charged portions of the molecules. Okay, so lipids, because they separate out from water, which of course is what makes up most of our body, these have a lot of functions, including forming barriers. Right, so we can see that automatically fats and oils are forming a barrier between outside the water and inside the water, and so we use these molecules to actually make up most of our cell membrane. Okay, we also use them as signaling molecules. So before I told you about the steroids and how they are mostly hormones. Right, so we also have some other signaling molecules that we'll get into here as well. And of course, you probably know about this the most. They are long-term energy storage molecules. That is, fats store energy for a long time. Right, so if you eat your candy bars sitting on the couch, first and foremost, that glucose is bound to existing glycogen molecules. If you don't get up and take a jog, over time, those extra glucose molecules stored as glycogen can be converted into fat. And so the fat can stick around for a long time so that we have energy in the event of a lack of resources later. This image down here on the right is showing you the different fat distribution of males versus females, and of course this is hormonally driven, where this is actually deposited. Because fat sticks around for a long time, so too do nonpolar fat-soluble compounds. So what I'm getting at here is that anything that is nonpolar, right, so it does not have a charge, it is soluble in lipids, not in water, these molecules are actually stored very readily in fat, as opposed to polar molecules, which are essentially used, right, and any excess are actually released in the wash, right, so they are excreted in the urine and or the feces. Okay, so whenever you eat a water-soluble vitamin, such as vitamin B, any excess vitamin B is going to be released out of your body via the efforts of your kidney and your intestines. On the other hand, there are 4 fat-soluble vitamins. So A, D, E, and K are fat-soluble vitamins that if you eat in excess of them today, they don't just go out with a wash. They are actually going to be stored in your tissues, right? So that's all great, right? So if you don't get out in the sun for a little bit, you still have some vitamin D stored in your tissues. So that's fantastic, hooray. Downside to this phenomenon is the fact that a lot of anthropogenic chemicals that we interact with, whether on purpose or not, are also lipid soluble, and a lot of these things are made this way so that they don't break down very readily in the environment. So things like pesticides, right? DDT is a great example, but also things like the chemicals in Roundup, right? For example, these are made, and you can see it on the bottle when you go to Home Depot and you buy weed killer. It says, you know, it lasts for 180 days. Well, the reason it lasts for so long is because it's lipid soluble, and so it's really hard to break those things down in the environment. Other things like brominated flame retardants, right? So in your carpets, in your desk chairs, in so many products that we use on a day-to-day basis, they have been treated with a flame retardant so that in the event of a fire, your furniture, your carpet, your house doesn't light up quite as quickly. But in the process of doing that, these chemicals ultimately leach into your body, and they're stored in your fat. They don't just go out in the wash. They are stored and they are accumulated in these regions or in your fatty tissue. And so there's tons of these different chemicals that you're interacting with all the time. The problem, of course, is that they stick around and they stay in places with concentrated fat. So, for example, in breast tissue, right, that is primarily fat, and so the chemicals that you're exposed to throughout your life are going to start accumulating more and more, and so these things can be pretty nasty, and they can mutate your DNA, and therefore they can lead to things like cancers. And so the reason, so I've given you a website here that explains the links, the very strong links between levels of these different persistent chemicals in breast tissue and abdominal fat and cancers. In the notes of this slide and that I gave you guys, there are also some other examples of some pretty nasty stuff that has happened because of these persistent chemicals ending up in your body. But on a lighter note, let's talk about the lipids. Right? So unlike the carbohydrates and the proteins and the nucleic acids, which we're going to see, the lipids don't form chains of monomers. They work a little bit differently. There are monomers, right? So we're going to take a look at those right here in this slide. And then the polymers are classified as 4 different kind of subclasses. All right, so first and foremost, we'll start with the easy one here. Glycerol, or glycerin, is a 3-carbon molecule, right, with hydroxyl, or with 3 hydroxyl functional groups attached, right? So essentially, glycerol or glycerin acts more or less like glue, and it kind of sticks together other types of lipid subunits. Right? What is a little bit more complex, and perhaps you've heard of before, are the fatty acids. Right? So the fatty acids, just like glycerin, just like any of the other forms of lipids here, are nonpolar, and so they do not have a charge. They have an equal sharing of electrons among all of the carbons, all of the hydrogens. For the most part, the fatty acids are simply chains of carbons with hydrogens attached pretty much anywhere that they can. Prime example of this are these saturated fatty acids. Right? So what you can see is a long chain, and this of course is a variable length. Right? You can have like 5 or 6 carbon long saturated fatty acids. You can have 100 carbon long fatty acids. Okay, so variable lengths. Take-home message here is that they are chains of carbons, lots and lots of hydrogens. For the saturated fats, saturated fatty acids in particular, there are single bonds joining together all of the carbons, and so what that means is that this molecule is literally saturated with as many hydrogens as physically possible, right? So if your clothes are saturated, they are full of water, they can't get any wetter. So here, these carbon chains are saturated with as many hydrogens as physically possible, and that is the case because of the single bonds. All right. On the other hand, we have unsaturated fatty acids. Okay, and so these are not completely saturated with hydrogens like the saturated are, and the reason for that is that there is at least one double bond. Okay, so you've heard of monounsaturated fatty acids, they have one double bond. Polyunsaturated fatty acids, they have multiple double bonds. Okay, that's not a distinction that we need to be familiar with, but you absolutely need to know the difference between saturated and unsaturated. Right, so here there are not two extra hydrogens. What we can also see in this image is that there are a couple different types of unsaturated fatty acids. All right, the cis is usually not included in these names, right? So cis is just understood, it's just the default. It's just understood because these are the naturally occurring fatty acids in the environment. Okay, specifically, cis unsaturated fatty acids are present in plants. Okay, and so so these are present in plants. Saturated fatty acids are also present naturally in the environment. So here we can see that the saturated fat shown here is found in butter. So saturated fats are generally found in animals. Unsaturated fatty acids are generally found in plant oils. Okay, and so even just the name kind of starts to distinguish the two for you. Saturated fats are solid at room temperature, and the reason for that is that they have these really nice straight carbon chains, which means that they can pack really closely together. As opposed to plant oils, they are liquid at room temperature. So we can see here that the double bond between the carbon and, the double bond between these two carbons actually puts a kink in the fatty acid chain. And what that means is that, instead of having the next fatty acid right here nice and close, this kink in the fatty acid chain makes the next fatty acid be a little bit farther away. And so this makes the chains not able to be as densely packed, and therefore they exhibit a liquid state of matter. Okay, again, as opposed to the saturated fats, which can have really closely packed tails. Okay, so those two are found in nature. We have enzymes to digest both of them. Look down at the bottom now. So here we have the feared, the hated trans fats. So it's actually trans unsaturated fatty acids. These have a characteristic in common with both the saturated and the unsaturated fatty acids. With the unsaturated, we see that there is at least one double bond. Okay. With the saturated fats, trans fats are also straight, or much straighter than the cis unsaturated fatty acids. Okay, so they have straight tails, which means that at room temperature, these things are solid for the most part. The reason for that is the positioning of the hydrogens. So what you can see here in the cis unsaturated fatty acid is that the hydrogens are on the same side of the double bond, or the cis configuration if you take an organic. On the trans fat, we can see that the hydrogens are on opposite or across sides of the double bond. And so that means that the tail is kept straighter. The fatty acid chains can be more densely packed. All right, so trans fats you can imagine as Crisco. Right, so it's vegetable shortening. Essentially, these do not occur naturally in nature. We can manipulate fats by adding heat, by adding catalysts, by adding extra hydrogens, taking hydrogens out, whatever. So we can make these things in the lab. We can modify all of these in some way. Trans fats in particular were developed to make foods able to last longer on the shelf, so more shelf stable. And the reason for that is that we don't have enzymes to break them down. or nobody does because it was just invented, right, within the last century. And so things last on the shelf longer. So when you go to buy tasty cakes at the store, at Wawa, they're filled with trans fats so that they can sit on the shelf for a long time. Secondly, trans fats, for better or worse, make food super tasty, right? They give food that nice flaky, buttery kind of texture, right? So this is something that was made in the lab for our modern diet choices. So glycerol and the fatty acids are true monomers of lipids. However, as I said, the lipids don't exactly join together in nice little chains like the carbohydrates do. Their molecules are much more diverse, and this actually gives rise to the fact that the lipids are involved in so many different functions. Okay? Every molecule that you see on the screen is a lipid, right? So it's really hard to see a pattern here. Let us walk through the 4 different types or subclasses of lipids: the triglycerides, eicosanoids, phospholipids, and the steroids. First, the triglycerides. These are meant for energy storage. These are made by both plants and animals, and they contain a glycerol. Right, so here is a glycerol. Remember, 3 carbons, right, and hydroxyl groups attached to each, and 3 fatty acids. So as we talked about before with the carbohydrates, glycerol can be joined to these 3 fatty acids via a dehydration or synthesis reaction, right? So if you took note on the last slide, the fatty acids also have a carboxyl group, a COOH. Given the action of a particular enzyme, a hydrogen and the hydroxyl can be removed, right, literally dehydrating this molecule to form, right, a triglyceride, right? So here is the fatty acid chain, here is the glycerol still, and here is that nice little oxygen separating the two. Okay, and of course water would be a byproduct, right? So when we as animals are making fat, we are essentially joining these fatty acids together with glycerol and storing them in our adipocytes, or our fat cells. Okay. When we need to use said fat, right? So when you start jogging a lot more, you need to start breaking down that fat. And so what then happens is that these molecules are decomposed, or they are hydrolyzed, right? So water is added back to the equation or to the molecule to separate the molecules out. Next, eicosanoids. These are another type of signaling molecules, but instead of being dumped into the blood to control faraway parts of the body, eicosanoids are local hormones. Pretty much every cell is capable of producing these, and they essentially help the cells to communicate with their neighboring cells and launch kind of controlled, coordinated responses to challenges. These eicosanoids, just like any of the other lipids that are found in your body, are made from whatever you're eating, right? So if you are eating lots of the reputably good fats like omega-3s and omega-6s, those fats, those fatty acid chains, are what are going to be incorporated into these molecules and ultimately into your body. Same time, if you're eating lots of trans fats, right, lots of Twinkies, lots of French fries from McDonald's, those trans fats are what are incorporated into these molecules. And we'll look at that again in just a second. For now, what I want you to see from the eicosanoids is that all of these very diverse molecules are derived from the same starting point called arachidonic acid. What we can see is that there are generally carbon chains that are, carbon rings that are not complete, so open carbon rings. Each time we see this little extra line here that is showing us a double bond. Sometimes the carbon rings are closed, right? Sometimes they're not. But what we can see here is that all of these molecules have some kind of open or closed carbon rings and attached fatty acid chains, right? So we've really only seen the carbons written out with all the hydrogens written out as well. Fatty acids can also be portrayed in this way as well, right? So Every angle, again, is a carbon, right? And whenever we see the extra line, that is indicating a double bond between the two carbons. Okay, so eicosanoids, open or closed carbon rings with two fatty acid chains attached. Okay, and so this family of molecules includes lots of different things, but what I want to point out are the prostaglandins. The prostaglandins are these local hormones that coordinate the inflammatory response and blood pressure in a localized area. So this really helps to heal, right? So to bring more blood to an area, so lots of white blood cells attacking things that might be invading an infected tissue. Inflammation kind of immobilizes an area, so you actually have time to heal without further damaging your tissue. All sorts of different things are involved in the inflammatory process, and prostaglandins in general regulate the inflammatory process. Some prostaglandins are pro-inflammation, some are anti-inflammation. All right, the next group of lipids here are the phospholipids, and these are critically important to our existence because these are what make up our cell membranes, or the majority of our cell membranes. Long word, but just like with anything else in anatomy, we can parse this word out a little bit. Phospho meaning phosphate. This happens to be another type of functional group. And lipids, of course, fatty acids here. Okay, and a glycerol even. So what we can see here is that a phosphate, which is a phosphorus surrounded by oxygens and hydrogens as appropriate. Okay, so this phosphate group happens to be polar. That is, it has a little charge here, and so it readily interacts with water. Okay, we have glue. Right, so this here is a glycerol, right? So it's literally gluing the phosphate onto two fatty acid chains, right? As we know, fatty acids are hydrophobic. So what we have here is a molecule that loves water up here and hates water. It's going to repel water down here. Okay, and so this configuration actually allows these molecules, and allows the membranes that they form to both interact with water and repel water and anything that is hydrophilic. Okay, and so what we can see here is a phenomenon that perhaps we've all witnessed before. Here is, right, lipids in water, as we've seen a couple times in this PowerPoint already. Just completely naturally, without any energy whatsoever, when you pour water or oil into water, these droplets are going to form, again reducing the surface area exposed with the water. And now if we add a little hydrophilic component to those droplets, so this little phosphate head, what happens, again completely naturally, no energy required, you don't have to have enzymes like constructing these little things. What's going to happen completely naturally is that all of the hydrophilic ends are going to face outward towards the water, and all the hydrophobic ends are going to face inward, right? And so this droplet is very stable. All right, so the water-loving part is interacting with water, a-okay, life is good. The hydrophobic areas are then protected by this hydrophilic shell. Okay, and so these little droplets that form automatically are called micelles, and our digestive system, a couple other things actually use them as well. In our cells, we use not only a single layer of phospholipids like we saw in the micelles, we actually use two layers. So we have a phospholipid bilayer, so two layers of phospholipids, to make essentially a little sandwich barrier that is selectively permeable. So it only selects certain things that can come in and out. All right, so the sandwich here, we can see the hydrophilic phosphate heads. Anytime you see this little sphere here, that is a phosphorus surrounded by oxygens. It loves water. Here is the second layer of phosphate heads. So inside the cell is mostly water. It's okay to interact with that. Outside the cell is mostly water. This layer, good to go to interact with water. And in between, we have a somewhat thick layer of hydrophobic areas, right? So essentially, the hydrophilic heads are going to prevent any hydrophobic substances from crossing into or out of the cell, and any hydrophilic substances are going to be prevented from entering or exiting the cell by the hydrophobic area. And so again, this allows the cell to be selectively permeable. It only lets things in or out that it wants, and does that by embedding proteins within the membrane. Okay, and so we'll talk about that more soon. All right, so before we move along to the last type of the lipids, I do want to take a moment to talk about trans fats. As I said before, more or less you are what you eat. So if you're eating lots of omega-3s, omega-6s, those fatty acid chains are going to be incorporated into your lipid molecules of your body. If you're eating lots of trans fats, those are the molecules that are going to be incorporated into your body. And so this becomes important, particularly when we're thinking about our cell membranes. Right? Remember that trans fats in particular have those straight tails. Right? So straight tails can pack really tightly together, and essentially they make a solid membrane on the outside of your cell, as opposed to eating plant oils. Right? That is eating vegetables, as opposed to just all straight-tailed substances. When you're eating plant oils, your phospholipids have these little kinks in their tails, and that essentially keeps the phospholipids apart so that your membrane can be fluid. And we'll talk all about the fluid mosaic of our cells, but long story short, it is critically important to be able to move your cells around, to be able to move stuff embedded within your cells all over the place. Okay, that is how your cells work. It needs to be that way. When you eat lots of trans fats, those straight tails are, of course, making your cell membranes chunkier, and so it's harder for your cells to move around, for example, this receptor. Okay, so your cell function is decreased when you're eating these straight-tailed molecules. Beyond that, the trans fats in particular are pro-inflammatory, right? So you might remember that animal fats also have straight tails, right? So like, why are trans fats any different than that, right? Why do they get such a bad reputation? Well, trans fats in particular lead to the production of more prostaglandin 2, one of those eicosanoids that happens to be pro-inflammatory. So the more prostaglandin 2 that is in your body, the more inflammation you're going to experience throughout your body. Okay. Also, to add insult to injury here, trans fats are going to decrease the production of prostaglandin 3. Prostaglandin 3 is made primarily from the omega-3s, and so prostaglandin 3 is anti-inflammatory. So normally prostaglandin 2 and 3 are balanced out to have just the right amount of inflammation, but when you eat trans fats, you have more pro-inflammatory and fewer anti-inflammatory molecules, right? So what that does is it leads to most of the normal everyday aches and pains that are exacerbated by inflammation, right? So lots more headaches, lots of skin problems, asthma, arthritis, these are all inflammatory issues. And so in general, you're going to be a lot crankier and unhealthy as you're eating these, right? In addition to, weight and all that other kind of stuff. Okay, over time, this additional inflammation that is systemic, that is throughout your entire body, can lead to high blood pressure because your arteries are clogged, right? These things stick together a lot, and it can even block your blood flow so much that you can have a heart attack, right? So that is certainly worse than eating saturated fats. Beyond that, the trans fats stick around a long time. As I said, these things were made in the lab. Therefore, we do not yet have an enzyme that is capable of immediately breaking these things down. And so, let's compare this to unsaturated and saturated. All right, so the unsaturated are found in plant oils. They have an 18-day half-life, so that means that the apple that you eat today, the pepper that you eat today, in 18 days half of the lipids that you acquired from those particular substances is going to be gone and out of your body, right? So really quick turnaround time relatively. Fully saturated fats, right? So when you eat a hamburger, when you drink a glass of milk, those fats, of course, we have been evolving with for a very long time, and so we also have enzymes that can quickly convert those saturated fats into unsaturated fats. And so we eat them, we turn them around into unsaturated, and again, we can cycle them out through our body relatively quickly. On the other hand, trans fats have a 51-day half-life, which means that in about 2 months, the French fries that you eat today, half of the French fries that you eat today are still going to be sticking around in your body, doing all of these lovely things like increasing inflammation and headaches and artery clogging and chunky cells. All of these things are going to be still occurring within a couple months. All right, so just a little bit of background. You might be interested in a little more research on that one. Okay, finally, types of lipids. Fourth and final subtype are the steroids. As we looked at briefly before in this lecture, the lipids, the steroids here, have overlapping carbon rings. Right? Might be 4, might be 5, might be 6, but overlapping carbon rings, that is, they do not have an oxygen separating these carbon rings. And again, that is critically important in distinguishing between steroids and carbohydrates. All right, the steroids can be hormones, which we'll look at here, actually right here. Testosterone and estrogen. We've seen estrogen before. I want to point out that cholesterol is the kind of starting point, right? The parent molecule for a complicated series of events, which we'll look at in this lecture as well, for the creation of steroid hormones like estrogen and testosterone. Estrogen and testosterone are one single reaction apart, and if you look closely, these are literally the same molecules except for one thing. Testosterone has no oxygen, or sorry, has no hydrogen down here, right? And so what we would consider to be a really big distinction between male and female is really a hydrogen, one single hydrogen. Okay. Other things that steroids can do is they can be pigments, they can be different types of vitamins, and of course cholesterol itself, even though it also gets a bad reputation, is critically important for your existence. Right? Not only is it going to become estrogen, testosterone, other types of hormones like aldosterone that are essentially regulating your blood pressure and how much urine output you have. Cholesterol not only becomes all those other molecules, but is also a really important part of our cell membrane. It increases the fluidity of our membrane so that when we go outside and it's really cold, our cells don't get more rigid, right? They're still fluid even when it's super cold. Okay, the third of 4 classes of biomolecules are the proteins. Okay. As I said earlier, the proteins have by far the most diverse functions, and the reason for this is that they are coded by our DNA to be incredibly diverse, so much so that they can bind specifically and tightly to their substrates. So they can recognize cellulose versus amylose, and they can tightly bind and efficiently catalyze reactions, for example. Right? So this is the key to the diversity of functionality in the proteins. They can bind specifically and tightly to their substrates. This chart here is showing you just some of the many different examples of what proteins can do. Of course, they can be enzymes, structural proteins, right? So they can, you know, they're part of our bones, they're part of our cells. Storage proteins, we can store energy in the form of amino acids. We can transport things, and in our next lesson we'll look at a cute little molecular motor that can like ship things around our cells. They can be hormones as well, receptors, so they are involved in receiving messages from other cells, other parts of the body. They allow us to move, right? So our skeletal muscles are made of a lot of proteins. And hooray, they are defensive proteins, so antibodies, which our white blood cells produce in response to exposure to a pathogen like a bacteria or a virus. These bind specifically and tightly to the bacteria or virus that they recognize. So exposure really is key to developing these antibodies. And when I say exposure, I don't mean necessarily being exposed to it like out in the wild, like going to the grocery store or something. Exposure can also be via a vaccine, which is simply the administration of pieces of a virus or bacteria or dead viruses and bacteria. All right, so the monomers of the proteins are called amino acids. They have 3 different components. 2 are functional groups, so amino acids. So we have an amine group, which is nitrogen bound to a bunch of hydrogens, and a carboxyl, which we've seen before, COOH. In this case, the H has dissociated, but that's okay. It now is polar and can interact with water. Right? Both of these functional groups are joined together by a carbon in the middle. The carbon is what attaches to this variable right here, the R group. And so there are 20 different types of molecules which can bind here, can bind where this variable is, and that R group is ultimately what determines the identity of the amino acid. Okay, so this here is an amino acid, right? So let me point out carbon, carbon, nitrogen. This is a telltale sign that you are looking at an amino acid. All right, carbon, carbon, nitrogen, and of course we have this double bonded oxygen up here. This is the R group, right? So something as simple as a hydrogen is one type of amino acid. Here again, carbon, carbon, nitrogen, the backbone, and this here, a lot more complex R group. Again, carbon, carbon, nitrogen, this is the R group, and this as well. So again, there are 20 different variants here. You are absolutely not required to memorize what these are, but it is important to know that of these 20, we can make about 12 of them. Okay, so we can eat whatever and we can synthesize our own. About 8 of them we absolutely have to acquire through our diet. And so humans are not unique in that. For example, your cat can never be vegetarian. It absolutely needs one of the amino acids found only in meat. Don't make your cat go vegetarian. It will not survive. Okay, just like we have seen with the lipids and with the carbohydrates, dehydration synthesis reaction joins monomers together. And so here again, carbon, carbon, nitrogen, here is 1, carbon, carbon, nitrogen, here is 2 amino acids. Water is removed, bond is formed, and so now we have a slightly longer chain, carbon, carbon, nitrogen, carbon, carbon, nitrogen, joined together with something called a peptide bond. And so we can keep on doing this. We can keep joining amino acids together. Again, note the pattern, carbon, carbon, nitrogen, chains, carbon, carbon, nitrogen. Seems like that's important if I keep saying it over and over again. And so when we join all of these amino acids together, we can clearly see the backbone here highlighted in green. And all of these little blue molecules here are the R groups. And so those R groups again are what determine the nature of the molecule. And this sequence, this order of amino acids is A, determined by our DNA. And our next lesson together is going to go into detail about how exactly that works. B, this sequence, right, this order of amino acids is called the primary structure of the protein. And that primary structure is one part of many in determining what the protein is capable of doing, what the function of the protein is. Okay, so again, the chemical nature of the R groups defines the characteristics of the protein. So for example, some R groups are charged, right? So a positive charge, a negative charge, and so in this big long chain of amino acids, we can see that the chain actually folds on itself positives attracted to negatives, right? And so what this image over here is showing you is each one of these is an amino acid, right? So carbon, carbon, nitrogen. And so this is part of a chain, part of the primary sequence, and here's another part of the chain, right? Might be far away, might be right next to each other, but either way, these two particular amino acids are attracted to each other. On the other hand, other amino acids will repel each other, right? Two negatives or two positives are going to repel each other. Okay, and so a different sequence would produce a very different shape. Okay, and so when we get this folding, we produce what is called the secondary structure. Right? So there's two kind of patterns that generally form. One is the alpha helix. Okay, again, carbon, carbon, nitrogen. Right? Carbon, carbon, nitrogen, and we can see these little R groups sticking off the side. We can also see that hydrogen bonds, our friends hydrogen bonds, are going to join together amino acids which maintain this structure. Okay. The second type of pattern or shape that is formed as the secondary structure is a beta-pleated sheet, which is what this is down here. Again, opposite charges are going to be attracted via hydrogen bonds, and so we can see this kind of accordion shape here that is maintained only by those hydrogen bonds. Okay. There's even more levels of structure here. So that chain of amino acids has been folded automatically into alpha helices and beta-pleated sheets. From there, the protein can further be folded, not all of them are, but can be further folded into a three-dimensional shape. And so again, this is very much tied to the identities of the amino acids in the sequence. Right here we can see that the hydrophobic regions of the protein are pointed outwards, so maybe this can be embedded within the cell's membrane. It can very readily interact with the lipid tails of the phospholipids, right? And at the same time, the hydrophilic regions right here are now pointed inward, and so now anything that is, or something specific that is hydrophilic, can actually pass through this little pore here and enter or exit the cell. Finally, there are some proteins, again, not all proteins, but some proteins can have a fourth and final level of structure called the quaternary structure. This is when multiple proteins, which each have their own sequence, their own alpha helices and beta-pleated sheets, their own three-dimensional structure, are actually squished together, right, and interact in such a way that multiple protein subunits come together to create the functionality of the entire protein. So a good example of this is the hemoglobin molecule. This is what makes your blood red. It is what carries oxygen, right? So these little iron molecules within each of the 4 protein subunits of your hemoglobin molecules can each bind to one oxygen molecule and dissociate from it at your tissues, therefore spitting it off so your tissues can actually use it. Other molecules with quaternary structure include some enzymes within your cells that are responsible for converting glucose into what is called pyruvate, or essentially starting to get some energy out of those glucose molecules. There are actually about 60 different proteins that come together to form the functional enzyme. So all of these are important structures for the function of the protein. This website down here helps you to essentially see all of these levels of structure. We can see the protein coming together. We can see the protein kind of melting down. So kind of interesting if you want to check it out. But for now, this image is showing you all of the different structural levels of a protein. Right, so here at the top is the primary structure. Each one of these tiny little spheres is representing an individual amino acid. This is the carboxyl end, and this is the amine end. Right, so presumably carbon, carbon, nitrogen all the way through. R groups sticking off the side, and ultimately the R groups and the carboxyl and amine portions actually can form hydrogen bonds to form alpha helices and or beta pleated sheets. Right, so note that in order to form these accordion shapes, there might be some amino acids that aren't part of that accordioning. Right, so very diverse structures and functions. From there, the same chain can be folded into a globular shape, so a three-dimensional shape, and we can even see within that the alpha helices and beta pleated sheets. Right? And finally, depending on the protein, sometimes the protein joins together with other proteins to form a full functional unit. It is ultimately all four levels of structure that determine the function of the protein. So I told you that proteins bind specifically and tightly to their substrates. In order to do so, all of these levels of structure absolutely have to be maintained. Okay, and so what we can see here is a functional enzyme, right? So molecule that is going to catalyze a particular reaction. The green chain right here is representing the primary structure, so a chain of amino acids. We can see that the chain of amino acids has been folded and that there are hydrogen bonds that are maintaining this structure. Okay, so maintaining this folding in such a way that ultimately these two amino acids are right next to this amino acid, producing a very specific key-like shape to this molecule. And again, that's only possible because the primary, secondary, and tertiary structures are maintained by these hydrogen bonds. When this configuration is maintained, this key-like shape is called the active site, and it is very specifically shaped to match this particular substrate. Right? So it's an enzyme, so maybe it's going to break the substrate down or it's going to join it together with something else. Either way, this is fitting together kind of like a lock and a key. Specific shape of protein fits with a specific substrate, and then and only then can this reaction be catalyzed. If the protein is denatured, right? So if it is unraveled by temperature or pH or whatever, what then is going to happen is that without those hydrogen bonds, this amino acid is now very far away from this amino acid, and so the active site, which again specifically and tightly binds to the substrate, is no longer going to be able to do that. So now we have an unfunctional enzyme. Okay. There are some proteins that are capable of unraveling and being denatured and then renaturing, but not all of them. Okay, and also some proteins can even denature and then form a completely different structure after they have been unraveled. Okay, and so what types of forces might denature a protein, right? So in other words, what conditions need to be maintained via homeostasis within the body in order to keep your proteins functioning and keeping your body running. Okay, so first example here is pH. Right, we talked about this a little bit before, but when we add extra hydrogen ions to a solution, right, so lower pH means higher abundance of hydrogen. This is going to disrupt the bonds that are already existing, usually the hydrogen bonds between these amino acids, and therefore the protein will be unraveled, and no longer do we have this very specific active site. Okay, another thing that can, or another factor that has to be maintained in order to keep your proteins intact is temperature. Okay, so when there is too much heat, this can also start shaking or vibrating your molecules a lot more, right? So more heat is more energy, means more movement of the molecules. And so this mechanical disturbance can also break those bonds that are holding the protein in its particular primary through tertiary structure. Okay, and so we've all actually seen this before when you cook an egg. The white of an egg is called albumin. It's actually a protein that is very abundant in your own blood, but also, of course, making up the egg white. In its natural, unadulterated configuration, this is a clear liquid, right? But as soon as you start applying heat, that is going to shake those molecules or vibrate those molecules so much that the bonds, as we can see down here, are going to break and the protein will be denatured. And the thing is, this particular protein, this albumin, is going to form different bonds when the original bonds are broken, right? So we actually get cross-linking between two sulfurs instead of sulfurs with hydrogens, and as a result, that protein is no longer going to be clear and liquid. It is going to be white and solid instead. Right? So ultimately here, all of the proteins have been denatured and cross-linked differently than the initial protein. And of course, as we know, once these bonds have been broken and then formed, the egg doesn't go back to being liquid and clear anymore. It's going to stay in this configuration. Okay, so again, critically important to maintain pH and body temperature in the human body, or else our proteins start to denature and are no longer functional. Okay. Not all proteins are created equally. So certain proteins are only able to maintain their structure at variable environmental conditions. So for example, the human body temperature stays the same, right? So all of our enzymes generally function best at the human body temperature. Some of them can actually increase their activity a little bit when you have a fever, so when you increase your body temperature just a little bit. But then, of course, too high of a fever, you lose that little edge and they start to unravel anyway. Okay. However, there are some enzymes, particularly for organisms that live in really hot, really cold environments. So thermophilic bacteria, for example, denature if they are cold, right? And so this is important to humans because we have actually been able to extract some of these enzymes and use them in something called PCR, polymerase chain reaction. And so we can actually sequence DNA, and we can do a lot of genetic research and therefore medicine development because we can use this heat-tolerant enzyme. Okay. Secondly, pH. Within the human body, your pH is not 7.4 everywhere, right? So in your digestive tract in particular, you have a lot of different specialized organs which have very different pHs, right? As you are surely aware, in your stomach, your pH is about 2. Your blood, the pH is about 7.4, and in your duodenum, so within your small intestine, your pH is more basic, so it's closer to 8. And so, as we can see here, there are certain enzymes that function best in these wildly different pHs. So this stomach enzyme called pepsin only functions in the stomach, right? As soon as food goes from your stomach into your small intestine, right, so it goes from this environment to this environment, the pepsin enzyme is going to denature, so it is inactivated, but in its place, trypsin, right, is going to be fully functional at this much higher pH, right? So like I said before, it is important to maintain the ideal pH conditions at different parts of your body, not neutral everywhere, right? Like you need your stomach to be 2 so the pepsin can work, but then you need your duodenum to be 8 so that trypsin can work and pepsin can stop working. All right, the third type of factor I want to talk about that affects enzyme activity, right, is the concentration of substrates and enzymes. So essentially, if you want to increase the rate of reaction or increase how much of a substance is going to be produced, you can either increase the substrate itself that you're working with, or you can increase the number of enzymes that you are producing, therefore making proteins of, that is in the appropriate area. Okay. A little bit more about enzymes because they're just so wildly important. Enzymes, for the most part, are proteins. There are a few examples where they are made of RNA. For the most part, enzymes are proteins. These enzymes are biological catalysts. That is, they catalyze reactions. They speed up the reaction. Yes, you can soak your pan in water, and that water will slowly but surely start decomposing the molecules that are stuck to said pan, but it is so much faster if you would have an enzyme to do the same thing. Okay, so enzymes, as we've talked about already, bind specifically and tightly to their substrate. So they have a very specific active site, which happens to match the shape of their substrates. Okay, and so the enzymes bring together these substrates and either add energy to the situation or put the molecules in such a position where it's a lot easier for the reaction to occur. Lots of different possibilities, but ultimately the enzymes push this reaction forward. Okay, once the reaction is complete, what we have is the product is release. Okay, so these substrates were joined together to create this product, and as we can see, the enzyme looks exactly the same at the end as it did in the beginning, which means that the enzyme is not changed, it's not used up by completing this reaction. It is just a machine, right? It's constructing or decomposing these molecules, specific molecules. Okay, so how exactly does it do that? Again, it can kind of put the substrates in such a position where it is easier for the reaction to occur, and so we can call this decreasing the energy of activation. In this image here, we can see a little bit more physics than we need to understand for this class, but essentially what I want to show you is that here are the reactants. They have relatively more free energy, so potential energy, and in order to get to the products, right, which have less free energy, so they are, in order to get from the reactants to the products, so the progress of the reaction, we have this energy requirement, right? So in order to make a little money, you need to spend a little bit money. And so this additional energy here is required in order to get the ball rolling, in order to get the reactants to interact in such a way that they form the products. Even in an exergonic reaction, or a reaction that releases energy. So what enzymes do, among other things, is that they essentially decrease the amount of money you need to spend in order to make money, right? So they essentially decrease the energy input, right, to this from this, of course, over here, in order for the reaction to proceed. Right? So take-home message here is that enzymes speed up the reaction by decreasing the energy of activation. Okay, and so if you are interested in a little bit more about this, I gave you guys a YouTube video down here if you want to hear more about that. Generally, our body has incredibly specific control over how many of particular products and substrates are actually available in our body. And so we do this by actually controlling the abundance of different enzymes in very complex, what are called metabolic pathways. So in general, we can start out with this, but maybe we want to control how much of this there is, and so we don't go right from A to C. We actually have different enzymes that regulate how much of each of these products are actually available. And so we've seen this already, right? So we've seen how amylose isn't digested immediately into glucose. Instead, there is an intermediate step. And of course, these metabolic pathways get so much more complex. Even this is a little bit simplified. What we can see here in the top is cholesterol, right? So this lipid molecule Cholesterol is processed by mitochondria to ultimately produce things like estradiol and testosterone. But we, of course, don't go right from cholesterol to estradiol. There are enzymes that regulate how much of each of these intermediate chemicals there are as well. And so some of these intermediate chemicals have their own functions, like progesterone or pregnenolone. And so, again, we can very closely regulate how much estradiol versus testosterone we have. And just a little bit here, the issue with BPA, right? So maybe a few years ago you heard this big debate about whether we have this plasticizing chemical within our water bottles, and essentially people said, absolutely not, this is ridiculous. It is feminizing our children, it is feminizing our adults even. Like, we need to get this chemical out of plastic water bottles. And so essentially there was a boycott, and lo and behold, the market actually had the power to get BPA out of water bottles. And so now you see BPA-free water bottles and so on and so forth. The reason why BPA was implicated as feminizing was that it affected the function of this right here. So CYP19, there are other chemicals that are around that we use all the time, especially now that we're all washing our hands so much, that affect this enzyme right here. And so essentially when you deregulate this, you are converting more testosterone into estradiol or estrogen, and so that will of course affect behavior development and so on and so forth. Oh, and just throwing it out there, BPA is not gone. It has been replaced by another plasticizing chemical, which might be doing the same thing. And B, it is still present in the linings of canned food. It is present in grocery receipts, and all sorts of different things. So take-home message here is that these processes are very fragile to chemicals in the environment, but also the good news is that we, as consumers, have the power to get these chemicals out of the products that we buy. Just one more example of a metabolic pathway. What this is showing us here is the cell membrane. See that phospholipid bilayer? And this here is representing the nucleus, also phospholipid bilayers. And so what we can see here, just very briefly, is that there is a receptor in the cell membrane that binds to a signal to ultimately stimulate the cell to make more of itself, right? So if you get a cut on your skin, you need to replace that skin. And so the way that we tell the neighboring cells, like, hey, you need to fill in this hole, right? This big cut, we need to make more of ourselves to heal, is, right, a messenger binds to this receptor. And so instead of just sending the message from the receptor right into the nucleus to make another cell, it's a complex pathway that essentially amplifies the signal and makes sure that every single piece of the puzzle is in place and made before cell replication actually starts, right? So this receptor turns on some of these, this guy turns on lots of those, which turns on lots of these. And so each time the signal ramps up and ramps up and ramps up, and ultimately this is going to lead to the production of all of these different proteins down here. And that's important because each one of these proteins is going to regulate or promote a different part of the complex process of mitosis. Right? So without all of these and without a really strong signal to get there, we don't have the ability to heal in the same way. One more note on this. This image is also showing us how in your skin, right, if we are exposed to UV radiation, right, so you're sitting outside and you get a suntan or you get a sunburn, that UV radiation has the ability to mutate particular genes, and if those genes happen to be involved in this regulatory process, what that can do is it can stimulate this cell replication process when it really shouldn't, right? So we haven't gotten a signal that we cut our skin and we need to heal it, but still we are getting this pathway stimulated, and as a result, cell replication is going to continue in kind of an uncontrolled manner, and it's going to make more and more and more skin cells that are essentially immortal, and therefore you have a tumor. Right, so that is just a very basic overview of how skin cancer actually happens. So the proteins, as I said, they do like everything in your body. Every single process has proteins involved, and it is of critical importance to have the primary, secondary, tertiary, and if applicable, quaternary structure intact in order for a protein to function properly. And what that means is ultimately that the DNA that codes for that protein has to be correct, right? It has to have the right sequence. The transcription and translation processes, which we'll talk more about in our next lesson, they also have to be completed correctly, right? Giving us the appropriate sequence of the protein. And finally, that protein has to be folded in the appropriate way, given different organelles. Again, we're going to get into that in order to produce a functional protein. If there is any kind of issue at any one of these steps, it leads to disease. Okay, and so each one of these diseases that's listed to the side, off to the right, and many, many more, are all linked to a problem with this step right here. So even one nucleotide difference in your DNA sequence can lead to a faulty protein, can lead to some really serious and often fatal diseases. Okay, finally, a fourth and final class of biomolecules, the nucleic acids. These you might know best as they are important in storing and expressing genetic material. So all of those sequences that code for proteins that do everything in your body is all stored in DNA. The nucleic acids are also important in as really ready energy sources, as we'll see here in a second, as well as some signaling molecules. So some of that like relaying of the information from the receptor in your skin down to the nucleus for gene expression, that is that involves what is called a G protein, which is a type of nucleic acid. These signals are also important in detecting hormones and, again, amplifying those messages. Okay, so specific nucleic acids, DNA and RNA. DNA is called deoxyribonucleic acid, emphasis on the deoxy, we'll get to that in a moment, and ribonucleic acid, the RNA. The monomers of nucleic acids are nucleotides, and each nucleotide contains 3 different components. First of all, there's a phosphate. Remember that this is a functional group. It is a phosphorus, a P, surrounded by O's for oxygen. We also have a 5-carbon sugar, and the 5-carbon sugar is one of the few ways that we can determine DNA versus RNA. A note about the 5-carbon sugar, it is, we can talk about this sugar and its 5 carbons by labeling them. And again, if you've taken organic chemistry, you might know this already, but we can name the carbons as 1 prime. So this little apostrophe here is prime, 2 prime, 3 prime, 4 prime, and 5 prime. And so this is important because this is essentially how we talk about kind of the top and the bottom of the molecule, not only the nucleotide, but like the entire strand of DNA or RNA. This side, the phosphate side, is the 5' end, so the top of the molecule. This side is the 3' end, the essentially bottom of the molecule. Okay, so we'll be using those terms, 5' and 3' end, a few times soon. Okay, the third and final component of each and every nucleotide monomer of the nucleic acids is a nitrogenous base. And nitrogenous just means that there's lots of nitrogens in these molecules. Okay, the nucleic acid types, so DNA versus RNA in particular right now, differ in the sugar itself and the nitrogenous base possibilities. Okay, so first of all, DNA contains a deoxyribose, whereas RNA contains a ribose. And so I want to point out that on the 2' carbon, all right, here's 5' at the top, here's 3' at the bottom. On the 2' carbon, there is a hydroxyl, right? So an OH on ribose, but on deoxyribose, this has been deoxygenated, right? So there's no oxygen there, but there is an oxygen on the ribose. And so sometimes, as we've seen before, the hydrogens are just kind of implied in these molecules, so sometimes you won't see anything on this carbon. Okay. The functional difference between these two structures involves how long we want these molecules to stick around. Okay, so deoxyribose, so DNA, is the code for all the proteins in your body, super important to keep around for the entire lifespan of the cell. On the other hand, ribonucleic acid, RNA, is a transient molecule. This is, essentially like a little post-it note, like you copy down a piece of the DNA, you use that little note for a little while, and then we don't need the post-it anymore, we just throw it away. And that's important. We don't want that RNA to stick around forever. And so in your cells, if an enzyme is taking a look at DNA and RNA, at these nucleic acids, if it sees an OH on the 2nd carbon, it's going to start breaking down the RNA, right? We don't want that there forever, so it's going to start breaking it down. But on the other hand, if it sees just the hydrogen, it is not going to break that down. Okay, so DNA gets to stick around a long time, and the way that we know that is because of this hydrogen right here. The second difference between DNA and RNA is what nitrogenous bases they can have. 3 of them, adenine, cytosine, and guanine, are found in both DNA and RNA. However, thymine only occurs in DNA, and uracil only occurs in RNA. And so these are those nitrogenous bases. You are absolutely not required to recognize each one of these, but you should be familiar with A, C, G, T, and U, right? Adenine, cytosine, guanine, thymine, and uracil. Okay. So those are individual nucleotides. As we know, a dehydration reaction can join together multiple monomers, in this case multiple nucleotides, into a larger polymer. Keep in mind that the top of the molecule always binds to the bottom of the next molecule. So here is the phosphate group, P surrounded by O's. Here is the sugar. Here's the bottom of the molecule, the 3' molecule, which is going to bind to the phosphate of the next nucleotide down. So again, here's the phosphate, here is 5' sugar, and these are the nitrogenous bases that are actually determining the identity of the nucleotide. Okay, so nucleotides are joined together into long chains. Right? So here we can see phosphate, sugar, nitrogenous base, phosphate, sugar, nitrogenous base. Each one of these is an individual nucleic acid. This is clearly the carbon backbone, and the sequence of the DNA is usually just referred to by the letter representing the nitrogenous base. Okay, so here a sequence of DNA, A, T, T, G, C, blah, blah, blah. The sequence of nucleotides is going to determine the message encoded by the molecule. So essentially, it is going to code for what amino acid sequence is going to be constructed to make a protein. Okay. Remember that even though this is usually what we see when we talk about the sequence of DNA, the nucleotide itself also includes the phosphate and the sugar. The nitrogenous bases themselves are complementary. That is, we can see that there are hydrogen bonds that join together only particular pairs of nucleotides. So these hydrogen bonds, as we talked about before, are what maintain the double helical structure of DNA. Also, RNA happens to only be a single strand of nucleotides. However, it often folds on itself, so one strand, but folding kind of like a protein does, joined together at these complementary nitrogenous bases. Right? So it actually forms a three-dimensional structure even though it's only one strand of nucleotides. These complementary bases are adenine and thymine. Right? So in DNA, A always pairs with T. Right? You usually see two hydrogen bonds between them. A always pairs with T, T always pairs with A. If you are looking at RNA, instead of a T, you will see a U. Right? So A pairs with U in RNA. Okay, secondly, cytosine always pairs with guanine, C and G, G and C, in DNA or RNA, doesn't matter. And again, you usually see three nitrogenous bases joining this together. Okay, so here we can see two strands of DNA. These green molecules here are representing the nitrogenous bases. We can see C's and G's bound by 3 hydrogen bonds, T's and A's bound with 2. What I also want to point out here is that the two strands of DNA are what is called anti-parallel. That is, let's see, phosphate sugar nitrogenous base, phosphate sugar nitrogenous base. This is the 5' end of the chain. Down here is the 3' end of the carbon or of the chain. Okay. If you look at this second strand over here, you will see it looks upside down because it kind of is. Okay, so here we see a phosphate. Here is the phosphate for this next nucleotide and so on and so forth. So this is the 5' end and this is the 3' end. So essentially these two strands aren't facing the same direction. They are anti-parallel. One is upside down. And we can see that over here, 5' end versus 3' end. And so this is important because always enzymes are going to start reading these molecules, either DNA or RNA, at the 5' end. Like the beginning, the top of the molecule is the 5' end. Okay, so to summarize, DNA versus RNA. RNA uses uracil instead of thymine, so only U's, never T's. RNA has an OH on the 2' carbon, not just an H. And finally, RNA is always single-stranded and transient, even though it often folds on itself with those complementary bases. So it looks three-dimensional. Okay, there are actually 3 different types of RNA, and I'm not going to go into these in too much detail now because in our next lesson together, we're going to talk about them in detail. So very briefly, RNA is used in protein synthesis, so essentially transferring the message from DNA to a protein. All of this RNA is encoded within the DNA, right? So it's just essentially a copy of the DNA with those slight modifications. The 3 types of RNA that we'll talk about are messenger RNA, so literally like the transcription of one particular gene in DNA. rRNA is a ribosomal RNA. This actually has enzymatic function and it helps to physically construct proteins using mRNA coding. And finally, tRNA, which is what I actually have for you pictured. tRNA is transfer RNA. It, again, is single-stranded, but it folds on itself with these complementary bases so that it literally looks like a T. That's not why it's called tRNA. It's called T for transfer. But essentially, these these RNA molecules transfer amino acids to the site of protein synthesis. So they're going to attach to the amino acid up here and attach to the mRNA down here. Again, we will get into that. All right, the final type of molecule that I want to talk to you about today is ATP, adenosine triphosphate. So tri for 3, phosphate for phosphates. This has 3 phosphates. It is a nucleotide because it has a nitrogenous base. It has a 5-carbon sugar, and of course 1, 2, and 3 phosphates. So P surrounded by O's. The bonds in between these phosphate groups, as you can see, is this little squiggle here representing a high energy covalent bond. And so essentially what this is showing us is that the phosphates automatically dissociate from their hydrogen ions within water, which, you know, our bodies are water, so automatically dissociate from hydrogen. And so what we have here are 2 negatively charged molecules that are super close together. If you've ever tried to put 2 negative ends of magnets together, you'll notice that they are resistant to being close to each other. They physically want to, or they physically push each other apart. However, the batteries, in this case the phosphate groups, are actually tethered together by this covalent bond. Right, so when this covalent bond is broken between the 2nd and 3rd phosphate, this phosphate, the 3rd phosphate, is going to shoot off, and energy is going to burst forth from this covalent bond. And so that little bit of energy is what is actually used to fuel our cellular processes. Okay, so just to elaborate on that just a little bit, up here at the top we have ATP. The high energy covalent bond that we are interested in is this one right here. It is storing lots of potential energy, right? As soon as that bond is cut, phosphate shoots off because negative hates to be near negative, and that energy can then be utilized by enzymes or whatever to catalyze reactions. And so in order to break the bond between the 2nd and 3rd carbon, we of course need to add water, and this is usually done by an enzyme, go figure. So this is hydrolysis. This phosphate is released, and a little burst of energy, which happens to be just enough to fuel most of our cellular processes, can then be harnessed. Okay, so this is an exergonic reaction. It releases energy. It is catabolic, right? We start with a bigger one and we make smaller molecules, and it is a decomposition, so hydrolysis or decomposition reaction. Okay, so what this image is showing you here is that ATP can use this tiny little burst of energy in a lot of cool ways, and we'll see this over and over again. But for now, you know, whenever this phosphate is released, right, therefore leaving ADP, adenosine diphosphate, in its place, this actually moves the little feet of what are called molecular motors to actually like ship things across the cell. We'll look at that in our next lesson as well. ATP can be used to physically change the shape of a protein pump. Right? So here, when the phosphate here is actually released, it can actually change the shape of this little purple molecule here and push stuff kind of uphill. Right? And we'll talk about that as well. Finally, chemical work. When the phosphate released from ATP binds to one of the reactants, sometimes it's actually a more stable configuration for the 2 reactants to bind together into the product than it is to keep this phosphate on the initial reactant. And so lots of different ways ATP works. Essentially, we need to know ATP because this is what fuels all the magic of our cells. And in its place, ATP releases energy, and we are left with ADP, adenosine diphosphate, and this inorganic phosphate. And so given the process of cellular respiration, we can actually recycle this molecule and put the phosphate right back onto ADP. Okay, so in conclusion, the function of these biomolecules, remember that the carbs provide energy actually to make ATP, support recognizing other cells. Lipids make our cell membranes. They help us to store energy for a long time. They signal nearby cells or far away cells. Proteins do everything, and nucleic acids store our hereditary genetic information. They are ready energy in the form of ATP. And finally, they can provide signaling between or within a cell for the most part. So thank you guys very much for watching this. My suggestion to learn these biomolecules would be to make a chart. So essentially make a chart with, all 4 of these classes. All right, have a function category, right? What do each of these things do? All right, and then literally copy and paste structures that I've given you guys from these lectures. So what is the shape of a monomer? Right? What is the name of the monomer? Monosaccharide, glucose, etc. What is the structure and name of polymers? Right? Any other special notes? And so by making this chart, you can have a really easy way to compare what these different structures look like for identification. Also, you can have this as a nice easy reference just in case you need to clarify what molecule is what. It would certainly be easier to look at one big chart than it would be to flip through dozens of slides worth of notes. And again, thank you guys so much and have a wonderful day.